Hi, everybody. It's a beautiful day, made even better because it's time for biology with Miss Judd. Now, it is a windy day today, so I apologize if you can hear the wind in the background, but being outside behind this huge and beautiful tree is the perfect place to talk about cell division. Because you ever thought to yourself, how did a seed, this tiny little thing, grow up to be this beautiful tree? Well, that has everything to do with cell division. It's the same reason that we get a cut on our finger and it repairs itself. Or it's the same reason that cells can die and be replaced with new cells. All of that has to do with one cell becoming an identical two cells. One cell becoming an identical two. And that's the cell cycle and mitosis. Now I'm going to turn to my friend, Miss Judd in the corner. Hello, everybody. Miss Judd in the corner here to talk about Unit 2, Lesson 2, the cell cycle and mitosis. Now, things are going to get pretty detailed here, so I need everyone to be in a concentrated space. Make sure you're ready for this. I'm also going to introduce a new friend of mine, Ghost Miss Judd, and she's going to help us with some of the images. Now, the cell cycle and mitosis, the first thing we need to kind of review is DNA moving into a chromosome. Now, why does DNA move into a chromosome? Condensing and pushing together our DNA into this chromosome prevents DNA from tangling and damage in cell division. So remember, DNA is linear and stringy. When you're trying to pull that apart and it's just like stringy, that would be super difficult. When it condenses down into this chromosome, it makes it a lot easier. Now, this chromosome hangs out in our nucleus, that cell organelle. It's an organelle that holds our DNA, which is our genetic information. And we have a lot of DNA. So DNA becomes this organized, condensed unit called a chromosome, which is made up of DNA and proteins. You want to think packaged DNA. So what this looks like, and you're going to add these to your boxes in your notes, is that my cell has my nucleus, and inside my nucleus are my chromosomes, which are made up of condensed down DNA. So you see my crazy long stringy DNA getting wrapped around some proteins and wrapped around each other to make that chromosome. Now that it's a chromosome, we can talk about why cells divide. Cells divide for three reasons. First of all, growth. From a baby to adult, we need more cells to grow. Second, repair. If you get a cut, you need more cells to fill in the cut. And then three, replacement. When cells die, they need to be replaced by new cells. How do we get new cells? Division. Fun fact, skin cancer, since our unit mystery is about cancer, is the abnormal growth of cells. They don't know when to stop making more skin, so it creates that tumor that pops up. Now, my cell cycle is a series of events from when a cell grows, prepares to divide, and then divides into two new cells. So we go through three stages. We go through interphase, which is the preparation, mitosis, which is when the nucleus divides, and cytokinesis was when the cell actually splits apart. And we're going to dive into those three parts, starting with interphase. This is the period of growth before cell division. So looking at this image that looks very similar to the one in your notes, letter A is G1. In G1, it's the first growth, 1G, of our cell. It's growing, it's copying organelles and proteins. S is when we copy or synthesize all of our DNA. So if we have two cells and we need the same amount of DNA in both cells because we're copying it, then we need to make more DNA. And then we have G2, the second growth. This is when it's preparing to divide. And all three of these are considered interphase. As we go through the cell cycle, we are going to draw each phase and record important events. Ghost Miss Judd here to say that we need to make some changes to our notes. Order of events goes interphase, prophase, metaphase, and then anaphase. We need to take out telophase and write anaphase. And then cytokinesis becomes telophase. And then at the bottom, we can write cytokinesis. So please make those changes to your chart. So I'm going to turn to Ghost Miss Judd to help us with our key that we need to add. When we diagram the cell cycle in mitosis, we're going to look at the movement of four things: DNA in the form of chromosomes, the movement of chromatids, and what a centromere is. So let's go ahead and do a simple key so we remember what each one looks like. DNA is stringy and linear, so I can do 3 different segments of DNA and it'll look kind of like a mess, but that's what we want because DNA kind of is a mess. Chromosomes, when they're replicated, look like an X. So I can turn my DNA into chromosomes. Now, each part of my chromosome has a chromatid. So this is one chromatid and this is the second. So when my chromatids separate, each half is pulled apart. Now, what keeps my chromatids together are my centromeres. So in my centromere keeps it together. So that is my centromere. So I have 3 centromeres with these chromosomes. 3 centers to my replicated chromosome. All right, let's start off with interphase. My important events here is that my cell is not dividing in interphase. DNA is replicating, it's going through G1, S, and G2. Ghost Miss Jed, can you show us how to draw this? In interphase, I have my cell, my nucleus is still intact, and my DNA has been replicated. So I have a bunch of replicated DNA on the inside. Awesome. Let's talk about prophase. In prophase, DNA is condensing down to make chromosomes visible. Spindles appear, the nucleus disappears. In prophase, I have a cell, my nucleus is disappearing, and my chromosomes condense. So I have the chromosomes from my key. In metaphase, the chromosomes line up the center of the cell. The spindles attach to the center, that centromere, remember, of each chromosome. If we look here, each chromosome has a centromere attached, and those spindle lines are attaching in. In metaphase, I have my cell. My chromosomes move to the center of the cell, metaphase, middle. And then those spindles attach to the centromere. So I'm going to draw my centromere. And then from the ends of the cells, I'm going to add my spindles because that's where they attach from. And they attach to those centromeres. In anaphase, the spindles pull the chromosomes apart. So my 2 sister chromatids move to opposite ends of the cell. In anaphase, my cell starts to look a little bit like this because my spindles are pulling those chromatids apart. So on the other side is the other half of that replicated chromosome that is being pulled apart by those little pesky spindles. And finally, we have telophase. Telophase is when nuclear envelopes reform. We have 2 new nuclei. The chromosomes uncoil, the spindle disappears. I don't have 2 new cells yet, but I have 2 new nuclei here. In telophase is when I get the new nuclei. So my cell is still connected, but my nucleus is reforming. And because my DNA is now surrounded by something to hold it, it can unravel. So it can turn back from a chromosome into DNA. Then we have cytokinesis. And cytokinesis is when my cytoplasm pinches, so the cell divides out, and I have 2 identical new daughter cells. And these 2 identical new daughter cells can go into interphase. And in cytokinesis is when we have 2 identical daughter cells. So this is when I have 2 cells with full nucleus, and inside are the same DNA that was replicated back in interphase, and these two cells can move into interphase. Now, when mitosis doesn't go correctly, that is cancer. Cancer is when mitosis goes out of control. In a typical cell, if it's damaged or mutated, it'll go through cell suicide or apoptosis. In cancer, which is over on this side, if there's a mutation or a damage, it still continues to divide like crazy. This out of control mitosis until you have a whole blob of cells. And that's called a tumor. So until a tumor forms, there's uncontrolled cell growth. Back to you, Miss Judd. Ghost Miss Judd here to take over and say, I am proud of you. You guys are doing great work. And don't forget to do your practice. Have a good day and make good choices.